Okay, so we are moving on to topic 5.4. This concerns non-Mendelian genetics, which, if you remember, Gregor Mendel kind of came up with these laws of, independent assortment, law of segregation. Um, these are kind of exceptions to that rule. And first, I want to talk about another famous geneticist here, another M geneticist. This is Thomas Hunt, or T.H. Morgan. And this guy worked with fruit flies. If you'll notice, he has these pictures of flies up here, and this is early 1900s. When he was working with these flies, um, typically they have a medium. These flies eat this, they breed, they have a larva, they hatch. And what he was doing is he would try to mutate these flies. He would give them chemical mutagens and subject them to UV rays and things like that. He's trying to get a mutation in these flies, which you can see some mutations up here. This is a vestigial winged fly, it looks like. This one, it's different mutation. Looks like these wings are off to the side. But before all that, there was one of the first mutations he found, and as he was kind of randomly analyzing these flies, was he found a fly that had white eyes. Had white eyes. If you'll notice, this fly has no pigment in its eyes. This is a normal or wild type. That's what we typically in genetics will call a normal fly. This is a wild type fly, and the one he found had white eyes. So he asked himself, I wonder what the basis for this mutation. I wonder how this works. Keep in mind, we kind of knew that there was this kind of these underlying things that pieces of information that affected the phenotype of an animal based on Mendel's work. So we knew that existed. We didn't know a lot more about how it worked, though. So Mendel's cross, if you remember, he took what he did, and he just found one fly like this, and he bred it with kind of wild type flies, and he noticed that all the offspring of those flies had red eyes. This should make good sense to you, right? If you'll remember, if I was to take a true breeding, red-eyed individual and cross with some sort of mutant, which in this case we'll assume is recessive, in your F1 generation, you would expect to get flies that all show the exact same phenotype, right? Same phenotype, same genotype, all heterozygous. So far, not a big surprise. And that's what he got in the F1 generation. All offspring had red eyes. This kind of assumes that you have a recessive mutation here. But in that F2 generation, he got something a little bit unique. He still had, and again, if you cross these in based on Mendel's laws, you would get, and I'll do this cross real quick to prove to you. He still got 75% that showed this this dominant trait. But he noticed that he only had males. Only males were showing this this recessive trait. And he wondered why. Why is it that only males are coming up wide-eyed, right? This doesn't seem to follow Mendel's laws. So let's see if sorry about that. So what he came to find out is this system worked a little bit differently. It's what's called a sex-linked trait. Well, this wide eyes in the flies is a sex-linked trait. Okay, so in the parental generation, and he did this through other rounds of experimentation and bat crosses and things like this. But as you guys probably know by now, in the parental generation, you had if you're a female, you are XX. If you are male, you are XY. And what he found was in that wide-eyed fly, he had a mutation right here, and this was a let's call it W, little W. Stop that. Little W. And he found that we're going to call the wild type for this gene just big W. I know, like that. So he had a mutant. This mutant displays the trait because if you have a Y chromosome as a male, you do not have an X chromosome to kind of cover up these recessive conditions that you might have. If you're a female and you have a recessive trait, you have another X chromosome, which means that you can cover up for a recessive trait with a dominant trait, right? Males are oftentimes more susceptible to certain things because of this. And that goes for us too, right? So he went ahead and bred these flies. And if you'll kind of work this out, let's work out this cross for the F1 generation, what we would see. And in this case, we have to use our, I'll put the female up top over here. And then I'll put the male down here. Again, W's are not the best letter to use probably, but X big W, X little w, X big W, X little w, X big W, Y, and X big W, Y. So just to highlight here, these are our recessives, just as the W is kind of an awkward letter to use. You'll notice that if you go through here and you see X big W, X little w, that is a female that is red-eyed. Now, she does carry the trait. She is what's called a carrier, right? A carrier, somebody that does not show the recessive phenotype, but does carry that recessive trait. Over here, we have another female carrier. So both of our females, all of our female, half of our population, all of the females carry the trait. All of our males do not have the trait because they have a Y chromosome in that second slot right there. So this is kind of what he saw. Now, if we were to take these and breed them back together, right? So we would be taking females that were X big W, X little w, and males, which are X big W and Y. We end up with X big W, X big W, X big W, X little w. Then you end up with X big W, Y, and X little w, Y. And if you'll notice, let's go back. So we have our parental generation was X big W, X big W, and X little w, Y. And our F1 was, I'm just transferring this stuff into our F1 spot over here, was X big W, X little w. And we were going to cross that with the males, which were X big W, Y. Right? And this last cross, this is what we get in our F2 generation. We notice that this gene kind of splits out. We still have, again, 75% of our flies are going to be red-eyed right here, right? And 25 will be white-eyed. However, if we look at this from a male-female perspective, we start to see this result that he got in the F2 generation up here. 25% red-eyed female, 25% also were red-eyed female, making for a total of a 50% red-eyed female. Of course, one of these would be a carrier, right? Let me erase this carrier so we don't get confused. One of these 25% cohorts would be a carrier. And then he ended up with half of the males, or 25% of his total population, being male and red-eyed. Makes sense down here. That's this guy right here. And 25% ended up being male white-eyed. 25% had the mutation. Again, keep in mind, this male does not have, this male that I've circled down here, does not have that X chromosome to cover up that recessive trait. So he just shows that recessive trait. Again, kind of a big disadvantage for males. And there's things like color blindness, which you might have heard of as a male, something that affects males more often than females. So this is one exception to kind of the way Mendel's rules kind of work. Another one, this is called codominance. Actually, incomplete. This is incomplete dominance. Incomplete dominance. Incomplete dominance is a situation in which neither allele is dominant over the other. So if you look, we still have two alleles over here, right? CR and CW. Well, these don't necessarily code for, neither of these is recessive to the other, right? If you have both CWs, you end up with a white flower. If you have both CRs, you end up with red. If you end up with a heterozygous situation with CR and CW, you're going to end up with pink flowers. So this is simply one of those cases where you don't have this simple dominant recessive situation, which Mendelian genetics kind of covers, these discrete traits. You have these intermediate phenotypes, which are kind of between a red and a white, and this is called incomplete dominance. So let's move on to something called codominance. This is where both alleles are fully expressed. You're going to see this in blood typing. So in the blood type system, you have three different alleles. You have big IA, big IB, and little I. And again, a lot of you probably know the different blood types are type A, type B, and type O, right? And these use three different alleles to reach these blood types. Of course, if you have, there's a chart down here that'll tell you if you are big IA, or big IA and little I, and in other words, you have that recessive, you're going to be type A. If you are big IB, or big IB, little I, you're going to have type B. If you have a combination of A and B alleles, IA and IB, you will be type AB. And if you were little I, you'll be type O. So if I said, what are the possible blood types that could come from somebody that is type AB, and I'll circle these. I want you to pause the video, maybe work this out on your own. Type AB and type O, what possible blood types could arise from that, and then what percentages? And I'm going to go ahead and work that out over here. So we're crossing IA, IB with little I. So you can set up a simple Punnett square for this. I'm going a little crooked here. Let's see if I can move this back together. So let's put IA up here, IB over here, little I here, and you end up with 50%, and hopefully you got this, 50% type A based on this, right? Because I have type A here, type A here, type B here, type B here. I'm just writing the blood types over in the corners so you guys can see. 50% type A because it's IAI, which gives us type A, right? And 50% type B, which is IBI, which gives us type B. So 50% type A and 50% type B is what you should have gotten from that one. So finally, the last kind of exception I really want to touch on here is polygenic traits. Polygenic traits. So one of the examples for this is skin color, right? Polygenic traits, if you the root word poly, you should kind of think of means many or lots, something like that, right? Genic refers to genes, right? Many genes. These are traits that are not controlled by one gene, right? And actually, most of the traits that make you up probably have multiple genes interacting. So it's never really as simple as for a lot of traits, for how they teach you freshman year when they say, oh, blue eyes, little b, right? Brown eyes, big B, little b, or big B, one of those two. A lot of them aren't that simple, right? Polygenic traits are very common, and skin color is just one of those, right? So we see a situation right here where you have a trihybrid cross. These are three genes interacting. And the darkness of skin results from how many of, let's say, a dominant version of these genes you get. So in this case, instead of putting letters up here, they're putting the number of dominants that are in each of these haploid versions of the cell. So all the way up to a total of 3. And if you cross 3 or 3, you have very dark pigmented skin. If you have a small chance from one parent of getting 3 recessive alleles and 3 recessives from the other, or dominant, depending on what genes you're talking about, you get very light skin. Of course, this is a very simplistic model. But you do end up having this curve of skin colors here where you have kind of this intermediate skin pigmentation is most likely. And these kind of corner cases here of the very dark or very light skin is much less likely. Now, I drew this number down here because this is just a random Google I did. Found that there are probably around over between 300 and 400 at least genes that are known to be involved in skin color. So imagine we're dealing with 3 here. Imagine the fact that you've got about, you know, definitely over 300 uh, genes that are interacting here. Imagine how massive that cross would be, and you can see kind of how you're more likely to have these intermediate phenotypes when it comes to skin color, and it'll seem more like skin color is a blending effect as opposed to a Mendelian effect. Um, so I think I'm going to stop there for this video. I do have to make another video over Mendelian genetics, but I will follow this video with that one. Okay, thank you for